So thank you very much, everyone. The Galien Forum is for me a very nice mark as we come to the last 2 months of the year, an opportunity to sum up what we have accomplished, the plans and thoughts we have for the future, and 2 more months to fix whatever is still left on our bucket list. I was very pleased, thrilled when the organizers asked us to have this panel concluding the day focused on COVID-19. While on one hand it represents some of the most dramatic progress of science in this century, at the same time we still are living with COVID, although in a new shape, where we all can meet here feeling somewhat safe and comforted by what teams have done. Paul's and my company represent 2 of the vaccine companies that made it possible to meet here. And of course, treatments allow for those that get infected to reduce the fear of ending up maybe in the nearby Mount Sinai hospital or having a really bad fatal outcome. I'm very privileged to have this distinguished panel. Paul BURTON next to me, Chief Medical Officer of Moderna. I feel a real closeness in the sense what we accomplished together. Yes. But to make that happen, there were many contributors that were critical. At the very end, I'll start with Peter Marks, who leads the CBER, the division that is involved at the FDA with complex biological treatment, vaccine, gene therapy, and others, who, as you know, played a pivotal role through this lengthy journey from early 2020 to now in providing guidance to colleagues like Paul and myself and our teams, and always balanced as a physician urgency to see progress for patients with the thoughtfulness and caution of a regulator. Next to Peter is Florian KRAMMER, one of the big contributors in science. Florian is a professor at Mount Sinai and contributed many important papers on immune response to COVID infections, early response to vaccinations, and overall characterization of the virus together with the Mount Sinai group, who I think was one of the most prominent hospital on the East Coast in driving science. With an eye for the future where science and new technologies come closer, we welcome Mikael LONGMIRE, who is a physician and technologist from the Stanford Palo Alto area, the place to go when we want to have optimism about breakthroughs into the future. And I hope she will give us a lot of optimism in the way we can do clinical trials differently, maybe more patient-centered, maybe faster, because what right now limits progress in this industry is the resource demand, the expense on patients, on sponsors, and still the rather large volume of data that may not be utilizing all modern technologies to in a more easy to analyze pattern could be available to regulators. Well, we have recognized that SARS-CoV-2 is a formidable foe to have, and clearly many have been exhausted by it. The new generation of kids growing up saw a rising need of isolation, mental health challenges in a society that didn't welcome them as normal to start college or high school. And the toll beyond subtle general description of suffering is that 1 million lives has disappeared in the United States. COVID has been the 3rd leading killer beyond heart disease and cancer, and it has decreased life expectancy in the United States by 3 years. Nearly 230,000 deaths in 22. So we're sitting here feeling comfortable, but for many other colleagues, people, co-inhabitant in this wonderful planet, it can be much more damaging. So we owe it to everyone that suffer and to everyone that is a patient of the future to take the lessons learned and make sure we incorporate them into the strategies for the future. In some way, we learned a lot about COVID-19, and never before you have opened your mail or looked at now open source servers, whether peer-reviewed or not. The dominant part was a completely new virus and disease that from nowhere became basically all the morning literature of many of our scientists. And the things that really, in the end, mattered for developing vaccines and therapeutic was actually understanding of 3 proteins of SARS-CoV-2. The spike protein, which is how it attaches to the cell, the main protease that became the target of the major used therapeutic, and the polymerase that it shared with many other RNA viruses. Now, clearly the science has now focused and the efforts mainly on the spike, and for us, Pfizer, at the polymerase. And the outcomes of this, the 2 mRNA vaccines, has now been deployed to billions of people across the globe. And I would assume that at least 9 out of 10 in this room that has vaccinated has been on either the vaccine from Paul's teams from Moderna or the Pfizer-BioNTech vaccine. So the mRNA technology, in a way, came to transform the response and kept Peter and regulators busy in trying to figure out its advantages and its unknown. The spike protein was under tremendous pressure on the immune system, but it was very dynamic, very plastic in its ability to adjust and create immune evasion. And colleagues like Florian working day and night trying to understand how it imprinted the immune system and how the various variants may drive hypersomatic mutations in the B cells, how it affects our B cells in the lymph nodes, the circulating B cell. Lots of very important science was piling up, but it felt like Omicron, the latest of the strain, was always a step ahead. It seemed to be able to use a tremendous alphabet of genetic changes to constantly recombine. To stay in a speed more similar to the threats of the virus, the paradigm from flu was, with data from Moderna and Pfizer, put into a paradigm that allowed new variant updates to happen based on accumulated experience on the mRNA platform on historical variants and a thorough set of preclinical data. We feel this was a courageous and now shown to be, after many people are vaccinated, from real-world evidence, it seems to be a safe approach. We're still learning how much increased protection may or may not have come compared to the original, but I think from Paul's and my conversations and what we have said publicly, we remain optimistic that it's a step in the right direction. You see many different science reports coming up with pace, and we would just caution to too fast draw conclusion. There will be much larger studies coming from both companies that I think will give us a deep understanding, because this is complex. Each of us have a different medical history. Peter reminded us in the warming up here how different comorbidities or age may affect publications. And I think we have seen how previous infections may bias the response dramatically. So all of that we learn. When it comes to treatment, clearly we were impressed and thrilled with a test and treat paradigm from the current Biden administration that simplified for those that were vulnerable and developed with risk factors, what we call the high-risk patients for COVID-19 disease, were put into a system that could, within a day, get the diagnosis and the prescription in hand and be benefiting from about a 90% reduction in risk for hospitalization. And actually, in our studies, 100% in reduction in risk for a fatal outcome. In contrast to spike, the target of nirmatrelvir, the active component of Paxlovid, did not mutate that much, and it was far more of a lazy enzyme. However, it was by intention that we deployed a drug in a bit like if you are going to put a stopper in a lock to protect the key from getting in, you need to mimic the key to a very high degree. Otherwise, someone may reorganize the lock and your stopper won't fit. And that's really at a simple high level where we used AI technology and decades of precision chemistry to make sure that the nirmatrelvir drug very much was a mirror of the target of the protease. And when that stopper went in, the virus had great difficulty to do its job and expand. That was just a brief introduction, and now we're going to take a glimpse in the future. Maybe it became too long. I got so excited about what has happened.
It was awesome.
So I'll hand over now to my panelists, and I'll start with a question. Let's talk about the future and pandemic preparedness. What are top on your mind for the mission of today and the futures? We start here and go from this direction to the end. Paul.
No, thank you, Mikael. It's great to see everybody here this afternoon. Look, I have a few ideas. Let me maybe touch on two. My colleagues here will want to pick up. I think, I've been in the pharmaceutical industry now for over 20 years. I was a surgeon originally. And thinking back to being a physician and what I do now, I think so much of it is about knowledge translation. Even the statistics that you very nicely rattled off, we're all immune to numbers now. So vaccine confidence, but misinformation and knowledge translation, I think, is one big bucket. There was a Kaiser Family Foundation survey done a little while ago where they ranked the top 8 misinformation facts about COVID. 2/3 of people in the world today believe the top 4, pieces of misinformation. I think that's translating into hospitalizations and deaths on a daily basis here in the US and around the world. So I think that's one big bucket. The other one I would say is thinking back to when the pandemic started, and Peter has talked nicely about this. This virus evolved fairly slowly. It had cycle times of maybe 6 months. We could keep up with it. But even think back to last Thanksgiving Day when Omicron BA1 entered the world. We've had BA2, BA45. I think there's even been, a 2.75 in there as well. So, and that's in, less than a year. I think if we as a world, we stand on a point where we have to rebuild the immunity wall. If we miss that now, keeping up with the cycle times of this virus and the evolution, I think, we could be in danger of getting outsmarted. So those are the kind of 2 big buckets of things on my mind.
Thank you. Michelle.
So, I think you highlighted some of the incredible progress that's been made, but also really what's in front of us. And I think that when we look at where we are today with the virus, we still don't have, a vaccine that prevents infection. We don't have a vaccine that prevents symptoms. And I think that when you look at the overall burden to society, it is not insignificant today while we all sit in this room together. And it's important that we not only look forward around how can we really ensure that our level of immunity in our communities is where it needs to be. In addition, we need to think about, okay, if in fact this were to evolve further, how would we re-incentivize or launch clinical trials for what we've already been able to do for net new approaches and be able to do that faster and even more effectively than we did the last time. Because the one thing we know about viruses is that they're really evolution at warp speed, and we can't predict the future around where we are going to end up in the pandemic as it moves into an endemic. So I think there are a number of challenges in front of us. It's about community immunity to put brakes on where we're at today, but then also thinking through whether it's this virus or the next virus, how can we combat these challenges to human health in a faster, more effective way, leveraging what we've learned today.
Florian.
So just to clarify, do you mean pandemic preparedness in general or this pandemic?
I'm thinking about SARS-CoV-2 and possibly relatives to come.
So I think there are major challenges ahead of us, but I think with SARS-CoV-2, of course, the situation improved a lot, right? The case fatality rate is way down. There's a lot of baseline immunity in the population that protects many of us from severe disease. And I think gradually the situation is getting better and better. We still have a lot of deaths. This is way above what we typically see even in a severe influenza season. And there are other challenges too, right? We were in the fortunate situation that we had a lot of drugs, specifically before Omicron, that could be used, right? Paxlovid is one of them, but there were a number of monoclonal antibodies. There are even drugs that you can give as prophylactics, right? Unfortunately, Omicron and now a lot of subvariants of Omicron are kind of becoming a problem with that, right? There are now subvariants out of Omicron that are increasing in frequency that completely circumvent neutralization by Evusheld. So that drug might not be working anymore in the near future. That leaves one monoclonal that can be used, which is bebtelovimab, and a new variant might basically outcompete that as well, right? So instead of having more and more options, we are actually, with these new variants, seeing less and less options, and that's, of course, a problem. I think that's a big issue that I see for the, let's say, coming 12 months. And right now, it's still not possible to really predict what's happening with these variants, and we still don't understand very well how they are generated and how we can stop them we can stop the generation of new variants, right? So that's kind of what I see as the big challenge in the next few months with SARS-CoV-2.
Thanks. I think in general agreement here. I think the challenge we've seen is how do you keep up with the cycle time of something that is moving as fast as this is going, whether it's a monoclonal antibody or the vaccine. And that means I think we're going to have to put a lot of effort into looking at the next generation of vaccines and to figuring out how we leverage all of our knowledge to be able to update, for instance, the monoclonals in a way that allows us to have a product that's useful for a reasonable period of time, because by their nature, it looks like the monoclonals are going to be really only briefly useful if this virus keeps evolving the way it's going. I think the other thing, though, that I would say that's come out of this is that we have to really be able to leverage all of the knowledge that we've gained and not forget what we've learned over the course of this pandemic. It is very easy, I think, just as we throw away our masks, to throw away a lot of the key learning that has come along with this, and we have to be careful not to do so. I think that the saying is you don't throw out the baby with the bathwater, and I think that's a danger that we have here.
Now a question to start with Florian, Peter, and Paul. If you think about the most surprising of the biology of SARS-CoV-2, what comes to your mind? And maybe also, are the things that should make us most fearful? Among those, there have been recent reports that suggest some of the new variants have three-dimensional structure that suggests they may have evolved in rodents and then moving back into humans. How should we look upon that?
Maybe I start. I think that rodent hypothesis is not really something that has a lot of evidence behind it. But in general, I think the biggest surprise that I got from SARS-CoV-2 was that we saw this quick evolution, right? So just to set this into perspective, influenza changes all the time, and we talk about antigenic drift. And when you have to update the vaccine, that's because you had one cluster transition, right? So the virus changed enough so that we think the vaccine needs to be updated. And now with Omicron, that happened with SARS-CoV-2. This is somewhat surprising because compared to many other RNA viruses that mutate but cannot correct basically mistakes that they make when they replicate, coronaviruses have a proofreading activity, right? So they mutate actually much slower than other RNA viruses. So it was not expected that they move so quickly. And the main hypothesis, what's happening here right now, is that people who are somewhat immunosuppressed support long-term replication of the virus, sometimes for months. And we have evidence, for example, here in the city that we have patients where the virus was replicating for 4 months, 6 months, and accumulating a lot of mutations. You might even get recombination of different variants in that person, right? And so that might be a main mechanism how these new variants are generated, specifically if you're talking about variants that have 10 plus changes, right? And so the question is, how do we stop that? And how do we treat these people who have basically persistent infections? How do we stop this from happening? And this might be happening out in hundreds of thousands of people globally. And not all of these people will see the new variant, right? But the chances are there. And that's the biggest surprise for me. We don't understand this well, and we don't know what our treatment options are and how we find these people and treat them and make sure that they don't breed a new variant. I think for me, from a scientific perspective, that was the most surprising finding.
Peter, you did work with immunocompromised people in your career quite a lot.
Yeah, I mean, this has been I think this is it's a very plausible hypothesis, and it's almost, I think, gone from being a hypothesis to being a fact, because we're just seeing this in enough different cases. And it's a challenge because, unfortunately, treating the immunocompromised with the antivirals doesn't seem to eliminate this always, or very often. It turns out that we're going back, this is like back to the beginning. In some cases, we've had to go back to things like convalescent plasma collected from in the area where that individual who is immunocompromised is recently, so that they actually get an antibody complement of what is, instead of a monoclonal, polyclonal antibodies. And it can seem occasionally, not always, to clear the virus there. So we're going to have to follow up on that. But it's really been, I think this has really been a problem, because this is a breeding ground. If you have the virus going on for 4, 6, or more months even in people, there have been reports of a year even, there's a lot of opportunity for these variants to spin off and then take off.
Paul, what do you think about the biology, virology, and maybe do you see a future of improved vaccination of immunocompromised, or you think that there is a need for different treatment modalities for those?
I think on the immunocompromised front, there's many millions of people around the world, right? Certainly here in the US, living with, either acquired or other forms of immunocompromise. I don't think we do a great job of clearly articulating to them the value of vaccination or the value of getting an infusion of an antibody periodically. We could do better there. I think they are fairly well protected with intermittent vaccination, and obviously Peter and his team have brought that into the labeling now as well. I'd say for the biology, taking a slightly different stance, and Florian mentioned the case fatality rate. When you think back to the early days of the pandemic, it was 14, 15%. I mean, it was very high. This is a virus that, if it wants to, can do real damage, right? We've witnessed that. But so why did some people, all things being equal, get it and survive, and some people died, when something is so effective pathogenically? I think that gives you a glimpse into underlying biology. George mentioned it earlier. If we could understand that, I think that would be valuable. And long COVID too, we'll probably talk about that. But you know, why long COVID is real. I think for many months, everybody thought, well, it's just people are tired and they're worn out and they don't like masking. Long COVID is very real. And so why does it manifest neurologically in some people very clearly as diabetes, cardiovascular disease, and the tidal wave that will bring? That's been really remarkable to me.
So, Michel, you spoke a lot about community immunity, and how do you see the future in light of how much we changed in the ability to work remote, interact remote? How can we capitalize those technologies to do clinical trials fast and more efficient, shrinking the world?
Well, I think, you know, it's kind of to provide a little bit of context. So our company provides a platform to connect people to clinical research. And we do that through this methodology called decentralized clinical trials. And you know, we had been pioneering this concept. I was a physician scientist at Stanford researching rare disease. And it was really through that lens I realized, like, not everyone can come to Stanford. We need to be able to connect patients to investigational therapies. So we started Medable, and you know, really it was an edge case prior to the pandemic because, of course, high stakes, you know, pharmaceutical companies are risk averse. It sounds like good in promise, but maybe not good in practice to be able to shift some of the data collection to people on their own mobile phones, in their own homes. You know, again, what is the quality of that data? Big questions. And then the pandemic hit, and suddenly there was really no viable alternative. And so the concept of decentralized clinical trials really went from an edge case to a main case by necessity. And you know, we had a huge demand for the use of our technology coming from trials that were already in flight prior to the pandemic to trials that then were started, you know, once the pandemic ensued. And interestingly, you know, what we saw was, okay, certainly this pickup or kind of level of interest will decrease once the sites or clinics open back up. But, you know, really within about 6 months, you know, my practice, for example, we were back seeing patients. We were doing it with the right safety precautions. But what we've seen in the use of decentralized clinical trials is actually the continued acceleration of the adoption. And so what we did was we partnered with the Tufts Center for Drug Development, and we said, you know, we don't know what the data will show, but we'd like for you to evaluate the 2 years of our data throughout the pandemic and look at, you know, is this net positive, is this net negative, what can we really learn? And what they revealed was that there was a 5x return on investment for phase 2 and a 13x return on investment for a phase 3 clinical trial that utilized decentralized clinical trial methodologies. And this came down to what we see as kind of 2 driving factors. And again, we're continuing this research with a much greater degree of kind of focus. But this was around cycle time reduction and protocol amendment reduction. We found this to be really fascinating. Why it makes me excited is, you know, as a physician scientist, now a technology entrepreneur, what I see as the opportunity in clinical development, to answer your question, will it become faster, will it become less expensive? I think yes, and the reason I say that is that when you look at the number of drugs that have been approved over the last decade, we really haven't moved the needle on that number. Now, when you look at, say, silicon chips that are in production in Moore's Law, technology fundamentally changed our ability to actually get the outcomes we were going after from a manufacturing perspective. Clinical development, the bottleneck is the clinical trial. Or getting new drugs to patients, the bottleneck is the clinical trial. So I am bullish that the results we're showing with Tufts are the beginning of a Moore's Law-like phenomenon of technology leverage applied to clinical development, which I would argue we should be able to take more shots on goal. I mean, the company is sitting here. The foundational science really is entering a golden age. I really believe that our ability to cure disease and to evolve or develop effective therapies has never been bigger. We have to attack the clinical development bottleneck with technology leverage, as we have in every other industry. We have to empower consumers and patients to be involved and to leverage this. And so to answer your question in a long-winded, but I hope data-backed way, I think that we will see leverage in reducing the timelines and reducing the costs. And I think that we'll be able to harness the incredible scientific opportunity to improve human health better than we have historically, in an ongoing, in a more kind of impactful way year over year.
Peter, how do you look upon this from both a physician and regulator perspective? is it an incremental evolution? Will it be big leaps? Because in a way you could say the technology will soon be there where you would get data instantly through a cloud and basically be surveying trials in real time.
Well, let me say this. I actually think that patients reporting outcomes from home, they're probably more trustworthy than a lot of the medical records where it's well known that copy-paste errors from physicians create medical records that are a lot of gibberish.
High five.
And I can tell you because we review medical records when we get single patient investigation of drug applications, et cetera, and copy paste. So getting data directly from patients should not bother us at all. I think the wearable technology has become something where it's really amazing to me how some people say, well, but that's a socioeconomic issue. There's actually more problems right now with the way we do trials in terms of socioeconomic status than with wearable or iPhone technologies because, I live in a part of Washington where the homeless people actually go around looking for plugs to charge their phones. And it's terrible, don't get me wrong, but people have, that's like if they're going to spend what little money they have that device. So there's ways to get the data, and I think we will, if we adapt to this, we may be able to have clinical trials that are much richer in terms of the diversity of our country and really get across all the different minorities and underserved populations.
And for you, Paul, and Moderna being one of the most active vaccine trial sponsors in US at the moment, how fast do you see that you're prepared for this? Do you think you will be waiting for it to happen? You will be part of the wave? Because like all new technology, it's about being there at the right time, not too early, not too late.
Yeah. You mean, Mikael, in terms of how we do new trials and
using much patient-centered, at-home recording, digital, direct from participant reporting, etc.
Look, I couldn't agree more with Michelle and with Peter. I previously worked at Johnson & Johnson, and we did a study there, two actually, one called Heartline and one called Chief. And Chief was a heart failure study where it went on during the pandemic early on, and we actually delivered, randomized people and delivered them a diabetes drug to their home, and they did a PRO on their phone. And, it went tremendously well. And I think this digitization, democratization, and decentralization is the future. Again, at Moderna, we built with a company called Evidation, 100,000 person community. And out of that, we were able to enroll 1,500 of them, and we mailed them a home blood draw kit from a company called Your Bio. And so they would take some blood at home. They did it 3 times. They mailed it in, and then we did antibody levels on them. And the results are fantastic. And you could not imagine even 5 years ago doing that. So I think an amazing thing that has come out of this pandemic is exactly that. It has revolutionized, and I wasn't here for Rob's talk earlier, but he's a huge fan and has been for years, of how do we truly do pragmatic, large trials, go after big, important questions, and address them quickly and meaningfully. And I think we're on that verge.
I think some of this is just a matter of having the vision to do so. Had we had the vision to put QR codes on the back of the COVID-19 home tests, we'd actually know right now how many people today actually have COVID-19, because after you take the test, you'd scan the QR code, and you could even take a photo of the lines. But we didn't have the vision to do that. And it's such a simple thing, which, we would, if this were selling widgets, they would know to do that. But we didn't do that for public health.
We need to get Washington to come to New York City restaurants.
Right.
Everyone has QR code, the little kitchen in the corner. But I'm happy that you are probably have lived in New York before and tested our restaurant. It's an opportunity for everyone in the room to ask questions here. We did a little bit of a warm-up round tour of the COVID-19 science and medicine. If there are any burning questions, I'll invite you to raise your hand here. Do we have a mic?
I can probably talk loud enough. Bill Gruber from Pfizer.
Here's the mic.
Okay, great. Bill Gruber from Pfizer. I wanted to pull on a thread that Florian attempted to start here, and actually probably directed first to Peter, and then anybody else can respond. And that is that one of the things that allowed, as challenging as it was and as disastrous as it was at the first, to actually pivot and get to vaccines quickly was pre-work that had been done by the NIH in looking at MERS and SARS, originally stabilizing that spike protein and then porting it over to SARS coronavirus 2. And so my question really is, in thinking ahead to other potential pandemics, which actually worry me a bit more than COVID-19. I think over time, we're going to manage this. But having seen that, we had a decade between SARS and then MERS and another decade, so there's an expectation that in another decade, we might have something else. So that's a long-winded prelude, but my real question is, what is the government doing? Because I think they're best suited to essentially prepare for a future, identifying those things that have pandemic potential, to bring them up to a point that one could then port the technology over to something related that actually does provide a pandemic. I'd start with Peter.
Yeah, no, it's a great question. I think there's a fair amount of dialogue right now about what we need to do next in terms of mapping what the next pathogens could be, and how aggressive we should be about going beyond just mapping on paper and possibly even going to the next step of having actually some work done to have candidates even thought of. Some of that will all depend on where things go with funding, because just like everyone else is fatigued with COVID-19, Congress is fatigued with funding for COVID-19 or pathogens in general, and we're ready to move on to something other than pathogen response. But it's not going to go away. It's kind of like global warming. You can kind of ignore it, but it's not going away. And I do think as much as we want to think that there's not going to be, we've had, it's like you've put in your dues for the pandemic of the century. The problem is the statisticians among us will tell us that next year the probability that we're going to have the pandemic of the century is still pretty close to the probability that it was before the pandemic. So that's the problem here.
Maybe I can comment on this as well. So, we had, if you go back for roughly 100 years, we had a 1918 H1N1 pandemic. We had another pandemic in 57 with H2N2, another one in 68 with H3N2. H1N1 came back. We had HIV. We had the 2009 H1N1 pandemic. In addition to that, we had two situations where it was really close, right? SARS-CoV-1, that could have easily become a global pandemic. And the MERS outbreak in Korea was actually close to go southways too. And so when this pandemic started, I'm a very positive person, right? I tried to see also the positive aspects, and I was thinking, now people will get it. Now we will start to really prepare for the next pandemic. And I was thinking Manhattan-style project, right? Maybe even globally, so that next time we would have a vaccine very quickly, we would already have drugs before the virus hits. And I have to say, I don't see any of this. And I think for the next pandemic, just from my point of view, we're actually probably worse off than with this pandemic, because now we have a sizable proportion of the global population that doesn't want to get vaccinated, that is against all kinds of measures. And, the next virus might hit next year. And the truth is, if you look at all of this with connectivity of the globe right now, with the growing human-animal interface, with habitat destruction, with global warming, these things will happen more and more frequently. We're not going to have longer intervals between pandemics. We probably have shorter intervals. And I completely share your point of view. What scares me really is the next one, and that might be something that has a case fatality rate of 60%, and not 1.5 or something in that ballpark. So I'm worried about that.
I'm actually most worried about one with something about 10 or 15%, because you can find something, once you have Ebola-like, once you have Ebola-like mortality, it's usually easy. The thing leaves so much destruction in its wake that you can find it and wall it off reasonably quickly. But the sweet spot is probably 5 or 10% mortality rate, and we were close to it with this. That would be really a disaster. Or it could be that it has a higher case fatality rate in older individuals and not so much in younger individuals. But it keeps me up at night because I worry about that even for this virus evolving further to a place where if it's able to overcome the modest amount of T-cell mediated immunity that people over 60, 65 have, we could be in a bad place.
One more question here.
Just a question regarding the regulatory pathway. Currently, I guess COVID variants don't differentiate between countries. So the issue is there is no national registry in the U.S., but also working across the board with other countries. It's a major issue right now, and we're always trying to do catch-up to the variants of concern. So, I mean, is there any mindset, moving forward between, I guess, the pharmaceuticals and the regulatory bodies to change this paradigm somehow in terms of the clinical trials?
I think, look, I would say, at least speaking for an mRNA provider, we at least have speed on our side. We went from the VRBPAC that Peter held back in June to a new adapted vaccine in 8 weeks. I mean, that would have been unthinkable, actually manufacturing it and supplying it at large amounts. I'd go back to that earlier point I made. I think we have a chance now, and if as a world we don't take it and we don't embrace it and we don't rebuild this immunity wall, then I think everything else that these guys just talked about could come to pass around SARS-CoV-2.
Paul, we heard Florian say, within the SARS family, we may be better prepared now because of the technologies. But if there is a pandemic, maybe building on Bill Gruber's, it may come faster and faster, 5-7 years rather than 10 with a new virus. Florian stunned me a bit by saying, I think the world is less prepared because of all the divisiveness, all the lack of commitment to a public health measure, all the disinformation. Any more uplifting comment from you or Michelle for us leaving late afternoon from?
Well, look, COVID was, we were talking about this earlier, COVID was a disease that was kind of foisted onto the world. It destroyed families, communities, and it came out of nowhere. Contrary to flu, which is kind of seen, which has a high mortality. I mean, flu is a bad disease, but it's sort of seen as just part of life, right? And so people would do something about it. There is so much resistance to COVID today, and I think we have to get over that. I would say, as I said, I'm a surgeon by training, and we have an adage which is, see one, do one, teach one. That's not always appealing to members of the public, but we have seen one, and we've had a lot of people go through one. And Florian is right. We had these near misses, but the world really did go through a lockdown, manufacturing multiple different strains, a whole new paradigm of regulatory approvals, everything that we've all done. So I do think if and when the next one comes, we've at least got this playbook to open up, which we didn't have before.
But we do have to get it right from a public health perspective, yes, which is we have to keep the public health separate from politics. Mm-hmm. Because, I mean, with all due respect, Scotland managed to get 90+% of its population. I mean, you have places that were successful by matter of fact understanding that these are public health measures and we don't get them. I mean, comments that, I'm not going to repeat them here to give them airtime, but there were comments early on during this pandemic which really politicized this and very inappropriately led to people thinking that this was not what it was, which is an existential threat for us that we all have to rise to the occasion to address and take care of each other. And I think that getting mixed up in politics was a disaster, and it's still with us to this day. And I mean, I just look at the vaccination rates, death rates, and the red versus blue. I'm a non-political, and so it really troubles me that there are places around this country where a lot of people died because of who they associated with, rather than because they made active choices based on a real benefit risk of the actual knowledge of what vaccination could bring them.
One question to Peter and others here is, we lived in a new era of social media during this pandemic, and there were many people that voluntarily became celebrity speakers, taking on them to represent big communities by themselves. And some, maybe intentionally or non-intentionally, spread a lot of information that had limited validity or reproducibility. Unfortunately, even physicians tend to like to be influencers nowadays. Instead of a design costume, they like to be able to speak about all the parts of medicine specialties that they not have practiced. So how much were we drawn into the fame of social media that allowed a lot of these problems to happen?
Much more than anyone is willing to admit. Okay, I have two 20-somethings at home who happen to be still living at home at the moment. And it was amazing how much one particular football player, was able to make a difference. And then there were a couple that, and what we didn't hear about are the dozens of NFL football players that were going and getting vaccinated, because they were. I did webinars with them. But you didn't hear about those guys. You heard about those that were not getting vaccinated. And you didn't hear about the bunch of rock stars who were all vaccinated. You heard about those that were not getting vaccinated. And so that message got amplified because we collectively, that's what we get all buzzing about. And I think, whether it's subconsciously or it's just the nature of human curiosity, it amplified that message ridiculously. I mean, it's really hard because there were a lot of football players that were proud of the fact that they were vaccinated, and it didn't matter.
Michel, a lot of these technological platforms that are positively changing the world came from the US West Coast. But apparently their pandemic preparedness wasn't at the very best. Any reflection, you that are a tech person, but also a physician,
Sure.
It's not many that are in that space. Any advice for the future? Do we need a conference where tech and medicine are more closely together?
I mean, if I just take a step back and think, okay, as a physician, what works, right? And I think then you have to determine in today's society, what does that look like? And ultimately, I think where we miss the mark is we're not meeting people where they are and understanding that culture and what they're hearing around them is their reality. And I can say I believe these things because I'm a scientist and a physician, but I grew up in a house of scientists and physicians, and I've only ever known science and objective or deductive reasoning. So I think that what we need to do in this moment is realize that the type of connectivity that the everyday person has and each generation has is very different than being informed by your physician as it was 10 years ago. And we need to be able to have a message and a community that is driving the objectivity and the science in today's world. And if we don't do that, we will fail. That's why we're failing. So I think that the social media platforms are extremely insidious. At the same time, they are what they are, and humans have a predilection that's a form of addiction. And we need to really understand that and enable our influence as healthcare providers, as people who care about the health outcomes. We need an effective way to penetrate that involves a modern technology ecosystem.
Very good. So there are two questions to the panel here. Please stay. The last one will be a mix of a comedy and a horror show. But as a warm-up for that, we learned a lot on the technology side. We learned how fast science can move in the academic community. in a collective way across the globe. From China to the US, papers were coming daily or weekly and giving tremendous space to new learnings. So 2030, if you extend from these technologies and this fast-moving pace of science, what do you think we'll be seeing when it comes to vaccines or interventions that capitalize on the pandemic of 2020 to 22? Will vaccines solve the HIV challenge? Will it be involved in cancer? What other major breakthroughs that were seeds planted due to the struggle in this pandemic will happen? Paul, you start.
I think...
What will Moderna contribute?
Yeah, what will Moderna contribute? Look, I think in general, the pandemic, again, to Michel's point, has brought a digitization of information to people, and it's democratized research and knowledge flow. So that's a good thing. So I think people are getting more health literate. There's a lot we have to do there, clearly, but I think that's a good thing. I have the zeal of the convert around mRNA, having only worked on it for a year and a half. It's an amazing thing because you take the mRNA, you put it in a lipid nanoparticle, you inject it, will be taken up with a cell, and if you're targeting something that's in a mitochondria or a nuclear pore, think about trying to do that with a small molecule, a large molecule, a lot of off-target stuff. If you can get the target you're going after, it's almost over with mRNA because you get it in the cell, translation, post-translationally modified, and it goes after it. So I think the pandemic, again, really has shown that mRNA as a therapeutic can be highly effective. It's very safe. And now, where else can it go? We have personalized cancer vaccines. We're going after rare disease, cardiovascular disease. The power of the platform, the speed and agility and the fidelity of being able to really manufacture a medicine potentially on demand for a rare disease is tremendous. So again, something good has come out of it, out of all the terrible stuff that we've been through. Some good things have come out.
And Michel, in your place, will Medable have surpassed Facebook in being a more productive community of bringing health together?
Human benefit, for sure. I think we already have. But I think, it's really interesting where we are with technology. There's just so much leverage, and I do agree that, I think with what Paul said, which is, we're at a point where people are more willing to harness that leverage. And one thing that really excites me as someone who looks at technology for leverage is, let's take, what artificial intelligence and say neural networks, just look at how they've revolutionized chess. In 2019, the emergence of neural networks in chess have absolutely changed the game of chess for anyone who plays. And grandmasters say this. I look at the human immune system. It is so complicated that we have not even fully unlocked, I think, an inkling of the potential of the human immune system. Companies like Moderna, what we're seeing in immuno-oncology, our application of technology to understand these unknown paradigms where we're just scratching the surface on the potential for how we can harness that to treat human disease, I think is phenomenal. And I think if we apply neural networks like we have to chess, to things like the human immune system and our ability to then unlock new stage gates of therapeutics, I think that we're entering an era where our science will absolutely astound where we could be in a decade. Now, again, I think the main bottleneck is the clinical trials, so we need to continue to apply leverage there because the inhuman part of the science needs to be able to meet the foundational science for us to make the impact to human health.
So, Florian, I think Michel challenge you that the paper you're writing 2030 will be published in Journal of Computational Sciences and be a new algorithm for the immune system, and you could close your physical lab and just have a dry lab. Is that what we should expect 2030 from you?
So I think that's all interesting, and I think it goes, goes in that direction. But I wanted to bring in a different aspect, what I would wish for 2030, and what I would hope is a take-home message for parts of the globe here. We have new technologies that can be used to tackle a lot of the diseases out there for which we probably could easily develop vaccines, but we don't do it because there's no money in it. And I think the other thing that a lot of countries, a lot of communities learned during this pandemic is that they are not getting primary access to vaccines, right? We have to be honest. Low and middle income countries had a really hard time to get vaccine supplies, right? And I think what that leads to, that's what I hope, and we see glimpses of that right now, is that we have more and more tech transfer and more capacity building in low and middle income countries, leading to A, capacity, local capacity, independent capacity to respond to these emergencies, and B, development of, and maybe local development of vaccines that are needed in these countries. And I'm thinking about things like Lassa virus vaccines, Congo hemorrhagic fever virus vaccines, and so on and so forth. So for me, the ideal development in 2030 is that low and middle income countries have the capacity to make their own vaccines and address the problems that they have in their countries locally. So that's what I would hope to see.
I do look forward to your papers 2030, but I think we need Florian to do a sabbatical at Stanford. Pete, I hope you take a little bit of humor end of the day here.
But I would say we're.
What will come in your cloud?
No,
what
no,
application?
I'm going to pick up on what Florian said. I think actually in 2030 we will be a lot better off if we're able to bring our manufacturing technologies into the 21st century with advanced manufacturing, where you could do what Florian's talking about, because you could actually imagine that with continuous manufacturing, you could make this automated enough that you could plunk down. Right now, you can have a 1,000-foot clean room that can be shipped around the globe, that you could imagine shipping those around the globe, and you could, if this were automated enough, make it so that you could produce these vaccines in country. So I think that would be, it would probably help us too, because the ability to rapidly produce vaccines in a more disseminated way around the country could be helpful in the next pandemic or for the next outbreak of some kind. So I think manufacturing improvements would be good.
Terrific. Paul, I think we have a lot of startups here on our right hand coming up. We need to keep an eye on them.
We're going to talk to them.
Now it's the final lightning round. Each of you have 1.5 to 2 minutes. So we're sitting here maybe end of 2023 in a similar panel. What would a really good 2023 look like, but also being, unfortunately, realistic? What would a really bad 2023 look like? Start with Paul.
Okay, so I think for bad, the simple answer for me would be that this virus did some Knight's move again in evolution and really picked up some pathogenicity. You know, if we had infectivity like BQ1.1 or XBB with Delta killing ability, I mean, that would be a bad day, right? And if XBB had come at the start of this pandemic, I think we would be in a different situation now. So I think pathogenicity is huge. In terms of good, I would just say, you know, we are investing $500 million to build a plant in the Republic of Kenya. We've announced that. We have others around the world, and we are fully aligned with what Florian and Peter said, that, you know, to be able to do a strain change, I think, won't be ready by 23, but maybe by 25 we'll be in business. But I would end where I started. I think it's all about, and it's on us really, a clear, simple, unified public health message on what to do. What really are the facts about COVID? How severe is it? What do you need to do? What are the pros and cons of vaccination? And arm people with the facts. I think that is what good would look like.
Michelle, good and bad.
Okay, well, I'll start with bad because I think that was a good idea. I mean, I think that, you know, when we look beyond the pandemic and what the human race has been through, including the pandemic, I think we're at a bit of a breaking point, and I don't think we could separate out, you know, the impact to humanity of the evolution of the virus in a more concerning way than where we're going today with further, I mean, we know that the transmissibility is up, but the mortality rates, I think that our case fatality rates, I think you layer onto that the economic concerns, you know, the risk of nuclear war, the energy crisis in Europe, and I think that, you know, it's just very concerning because humanity can only, I mean, we endure so much. I think even in the pandemic, we're like, wow, how does this get worse? So I think, a bad 2023 is a combination of factors that ideally we're in a position today we can take a proactive position on and manage in our favor. I think there's still a lot of uncertainty. I think a good 2023 is a much different thing. It's taking the energy, the optimism, the 4-minute mile we've ran in a sense with the vaccine approvals, and also looking forward to, a world which has a lower risk of nuclear warfare and an energy crisis that I think we take an opportunity to take a much longer term view on how we're going to solve that full stop with, in my opinion, the use of things like, a better approach to nuclear energy as a solution to this. So I think a good 2023 is harnessing a hard moment in time to make real progress as the human species.
So I'm going to answer that strictly from a virological and immunological point of view. I think a good outcome would be basically a lower attack rate of the virus, fewer cases next winter. I mean, there will probably be again a winter season, but if the attack rates would be lower, if we have higher immunity in the population even than we have now through more vaccination and readily available antivirals, I think that could also reduce the evolutionary change of the virus and would allow us to have a relatively okay season with maybe less impact of COVID-19 than we see with influenza. That's the optimistic outlook. The less optimistic one is, of course, as we already heard, more immune escape and maybe a backbone that actually drives or is again more pathogenic than the typical Omicron backbone. And that, in my opinion, would be a problematic year next year if that happens. So in one scenario, the virus is less pathogenic, less problematic than influenza. In the other scenario, it stays above influenza. And that's what we see right now, so we'll see how that develops.
What odds do you put on the two scenarios?
50-50.
So you've left it to me to be even more horrifying. So I think on the bad side, it's that we have an evolution of further evolution of the variants, but could make it even worse if we have a bad flu season and RSV continues its badness. We could make it very confusing and very hard to sort out for providers, and that could actually make treatment difficult because giving the wrong antiviral for a given condition will not help things. So that could be a problem. On the upside is it could be that after people start coming down with COVID left and right during Thanksgiving, they're scared straight. They start to go and get their boosters, and people start to realize that we have to get on the same page and start working together to try to bring this collaboratively under control in the United States and globally, and that we work with our global partners to try to deal with this issue globally, because as Florian already mentioned, it's just an airline. I mean, it doesn't matter. It's just if a new variant emerges somewhere in Africa or in Asia, it's an airliner away from here. So I think if we can get to, that would be a good outcome, that people get vaccinated, because they're scared straight. And it's possible it could happen. I mean, it's also possible that you're going to levitate out of your chair now. That would be a good outcome.
Okay, I want to thank, first of all, the panelists. They made it possible. I deviated quite a lot to keep you excited from the grid questions and tried to put in a little bit provocative pieces, but they were just outstanding. So I'm very impressed and grateful. And to build on the two scenarios here, I really liked where Michelle was going. Right now, it's a frightening world, whether you're a virologist, a climate change scientist, or someone involved in understanding humanities and the dilemma between human learnings from terrible conflicts around the globe. Hopefully we'll see not just the rise of science, but the rise of the power of the people to come together and create a good 23 but I think we all are leaving here with quite some concern that we may need to wait longer. Thank you for staying with us today.